Hello everyone! Good day! Welcome back to my channel! If bago ka pa sa aking channel, please don't forget to click the subscribe and huwag kakalimutan i-click ang bell button para updated ka sa aking mga bagong videos. Okay, so today's topic is all about microbiology or bacteriology. So we will be talking specifically about microbial growth and microbial control. Today's objectives: at the end of this lecture, the student must be able to identify the different nutritional requirements for bacterial growth. So ano ba ang mga nutrition na kailangan ng mga microorganism para sila ay mabubuhay? Enumerate different methods of controlling microorganisms. List down organisms with specific nutritional requirements. Ano bang mga microorganism na may mga specific requirements? Gusto ba nila ng malamig? Gusto ba nila ng mainit na lugar? Okay, so let's start. Microbial growth. So as you can observe to our picture, at the background picture, you can observe it here, the growth of a certain microorganisms in a certain culture media. It increased in number of cells, but not in cell sizes. Unlike humans, from baby, we have a very small baby. And habang tumatagal, ang human, lalo silang lumalaki. But, as you can observe to our quote here, it increases in number of cells, not cell size. So, your microorganism or your bacteria increases in numbers, but not in cell size. Okay? So, the requirements for growth, we have the physical environment requirements, and of course, we have also a nutritional requirement. For your physical or environmental requirements, your microorganism needs a pH. Okay, a certain requirement. So, ano bang kailangang pH ng ating microorganism? Temperature, oxygen, carbon dioxide. While your nutritional requirements, you need carbon, nitrogen, water, salts, trace elements, and organic growth factors. Okay, let's talk about the physical requirements. When we talk about physical requirements, let's talk first about the pH. The optimum pH of an organism, okay, so they need to have a neutral or slight alkaline pH to survive. Okay, so most bacteria grows between the pH of 6.5 and 7.5. Molds and yeast grows between 5 and 6 pH. There are also some organisms that are acid-loving bacteria at a pH of 3.0, just like the tomato juice agar. And we have also an organism that they love alkaline-loving bacteria at the pH of 8 to 10. So, we are using an alkaline peptone water, just like your Vibrio cholerae. So, there are different temperature needed. So, we have, as you can observe it here, okay, as you can see to my mouse pad, so there are certain temperature that we call to a certain bacteria. If they want or if they live in a negative 10 to 20 degrees Celsius, we call them as a psychrophile microorganism. We have also the 0 to 30 degrees Celsius, the psychotropes. Then, to 10 to 50 degrees Celsius, the mesophiles. The 40 to 70 degree or 75 degrees Celsius, the thermophile microorganisms, while those organisms lives in a 70 to 110 degrees Celsius, very hot temperature, we call them as hyperthermophiles. So let's talk some organisms. Let's learn some organisms. What are a typical organism lives in this temperature? The psychrophilic or the cryophilic organisms, they are the cold lover organisms. So they grow between 0 degrees Celsius and 20 to 30 degrees Celsius. They are the first or the primary causative agent of food spoilage. So example of this are your Listeria monocytogenes or Listeria monocytogenes or monocytogenes. So these are a typical example of your psychrophilic or the cryophilic organism. They are a cold lovers. And of course, the Yersinia enterocoletica. As you can observe it here, okay, you can observe it here, the cyclotops. So, there is a danger zone, okay? So, the rapid growth of bacteria, some may produce toxins. So, at the temperature of 20 to 50 degrees Celsius, in this temperature, mapapansin nyo guys, na in this temperature, maraming mga organism ang mabubuhay. Okay? So, this is a very typical temperature para sa kanila. Okay? Yun. So, next is the mesophilic or the moderate temperature. Most pathogenic bacteria are mesophilic. Please take note, don't forget that most pathogenic bacteria are mesophilic. Pathogenic meaning it may cause harm to our body. They live in a 30 to 37 degrees Celsius. Next is the thermophilic or the warm temperature, 50 to 60 degrees Celsius. And the hyperthermophilic, the microorganisms lives 80 to 110 degrees Celsius. Next, some organism needs an oxygen. Okay, most of an organism needs an oxygen. So we call them as number 1, obligate or strict aerobes. They grow only in the presence of oxygen. When an organism grows only in the presence of oxygen, they are called to be an obligate or strict aerobes. So they need oxygen to be alive or to be living. Okay? So example of this are Pseudomonas aeruginosa, Neisseria, and your Brucella, and Bordetella and Francisella. Next is the obligate or strict anaerobes. Grows only in the absence of oxygen. They will not alive or they will not be living when there is a presence of oxygen. By the word itself, strict anaerobes, meaning absence of oxygen. So they are obligate or strict anaerobes. They grow only in the absence of oxygen. Example of these are bacteroids and Clostridium. Okay. So next is your obligate or strict anaerobe. So how does they live? Okay? Paano ba yan sila mabubuhay? Okay? Paano ba yan sila ibubuhay sa isang laboratory? By the use of anaerobic jars, and of course, we have also a disposable anaerobic jars, bags. Okay, so this is an example of anaerobic changer. You will be dispensing your microorganism here in a closed area. So this is an example of anaerobic chamber. So there will be no oxygen presence here. Okay, so as you can observe, it is a glass-proof environment. So wala talang oxygen na papasok dito. Okay, so this is a typical example of anaerobic jars. Okay, so this is a very tightly closed container. Okay, this is anaerobic indicator, the methylene blue. So dito natin malalaman kung wala na talagang oxygen pag magko-color blue siya. So this is your Petri plate. So yung lahat ng mga Petri dishes with microorganism with a Petri dish na mayroong organism, nandito sila. And of course, envelope containing sodium bicarbonate and sodium borohydride. So that releases carbon dioxide. Yun. Okay. So next, we have also a facultative anaerobes. So aerobes that can survive even in the absence of oxygen. So even without oxygen, some organisms may also live. Okay? So they are bahalag walay oxygen, kahit walang oxygen, mabubuhay pa rin sila. Okay? So they are called to be a facultative anaerobes. Example of this are Staphylococcus, Streptococcus, and Enterobacteriaceae. Next, we have also an aerotolerant anaerobes. So they are anaerobe that can survive even in the presence of oxygen. So aerotolerant anaerobes, kahit they are anaerobes, but when there is also an oxygen, they can also survive. Okay? So example is Enterococcus faecalis. Next, we have a microaerophilic organism. So it requires 2 to 8% oxygen. So it requires very small amount of oxygen. Example is your Campylobacter. Oxygen, carbon dioxide. So Topophilic, they need an increased carbon dioxide. We called an organism an increased, they need a very high amount of carbon dioxide. We call them as a capnophilic. One typical example or a typical example of capnophilic organisms are the following. We have the Neisseria, Haemophilus, Actinobacillus, and Cardiobacterium, Echinella, and Kingella. Okay, capnophiles require high carbon dioxide. So this is a very typical example that we are using in the laboratory. We use a candle jar. This is the basic example. We use candle, no, to produce carbon dioxide. Okay, so glass jars, this is just a jar, typical jar. Petri dishes with solid media, inverted, yan. And tubes with liquid media. You can also tubes, okay? So candle as a source of carbon dioxide. Okay. Next is we have also a carbon dioxide pocket, petri plates with bacterial culture, and the gas generators. So oxygen requirements, bacteria with oxygen requirements, we call them as obligate aerobes. They need a lot of oxygen. We have the facultative anaerobes, obligate anaerobes. So, dapat wala talaga silang oxygen. The aerotolerant anaerobes, some organisms may live even without with a small amount of oxygen. Microaerophiles, they need very small amount of oxygen to survive. Okay? So, next is nutritional requirements. Bacteria or microorganisms needs carbon. So this carbon, the purpose of carbon is for a structural organic molecules, energy source for synthesis of cellular components. Chemoheterotrophs or organotrophs uses organic carbon sources. We have also autotrophs or lithotrophs uses inorganic carbon dioxide. Please don't forget this. Organism uses organic carbon sources. We call them as chemoheterotrophs or organotrophs. Organotrophs or organic, right? Autotrophs or lithotrophs uses inorganic carbon dioxide. Organisms also needed nitrogen because the function of nitrogen is for the synthesis of proteins, RNA, DNAs, and ATP of a certain microorganism. Most bacteria decomposes proteins. Some bacteria uses ammonia or nitrate, and a few bacteria uses nitrogen and two in nitrogen fixations. Water or moisture. So this is used for a solvent for food. Okay, so your organism needs carbon, nitrogen, water, or moisture, as well as salt. Okay, so we have an organism also needs a lot of salt. We call them as a halophilic organisms, or they need high salt concentration. Please don't forget that an organism needs high salt concentration are called to be halophilic organisms. Example of this are your Vibrio. Next is mineral or elements, just like your magnesium, iron, potassium, and calcium. So inorganic elements requires in small amounts, usually as an enzyme cofactors. We have also a sulfur. Some microorganisms need sulfurs because your sulfur is needed in an amino acid, thiamines, and biotin synthesis of a certain bacteria. Most bacteria decomposes proteins, and some bacteria also uses sulfur or H2S. Next is phosphorus. Phosphorus, the function of phosphorus is for the synthesis of DNA, RNA, ATP, and membrane. Phosphorus is a source of PO4, phosphate, no, is a source of phosphorus. Okay, so let's talk about the bacterial growth or phases, or the bacterial growth phases. Okay, there are four stages in a bacterial growth. We have the log phase, the log phase, stationary phase, and the death phase. Let's talk about the log phase. In this phase, the log phase, or the period of adjustment, there is no lethal bacterial multiplication. Why? Because in this area or in this stage, the log phase, you can observe it here. It is very small curve. Okay? In your log phase, there is a very small curve because in this stage, the bacteria still adjusting. Okay? Nag-a-adjust siya sa kanyang environment. So, kumbaga, kumukuha pa siya ng kanyang food or nutrients sa kanyang or sa ating culture media na ginamit. So, there is no or little bacterial multiplication. So, wala pang mga multiplication na nangyari dahil in this stage, dito pa nagkumukuha ang ating mga microorganism ng kanilang mga food or in a preparation for a multiplication. And next, in a log phase, you can observe it here or also known as exponential phase. Okay? Bakit tinawag siya exponential phase? Dahil sa log phase natin, dito sila nagko-collect ng food. Then after that, since mayroon na silang enough food, ready na sila to multiply. So, exponential phase or the maximum multiplication. So, marami na sila. So, because they have a lot of energy to multiply, so in a log phase, dito na sila nagmo-multiply. Most metabolically active, most susceptible to antibiotics. Yan. Or antibiotics. Next is your stationary phase. Since marami na ang ating microorganism, okay, marami na sila in a lower log phase. Sa stationary phase, as you can observe, sa karami-rami nila, dito na nauubos ang mga nutrients na nandito sa ating culture media or sa ating culture medium. So dito, nagpa-plateau na. So wala nang growth na nangyayari dito dahil naubos na ng mga microorganism ang isang or ang food na nandoon sa ating culture media. So, in your stationary phase or the plateau phase, okay, the number of living organism is equal to the number of dead. So, there is a lack of nutrients dahil marami na sila. Just imagine a certain population. So, kung mayroon lang kaunting supply ng food, so mauubusan na tayo ng food. So, yun din nangyayari sa ating mga microorganism. Naubusan na sila ng nutrients. So, there is an accumulation of toxins. So, when there is an accumulation of toxins, there is a lot of dead microorganism. Okay? So, there is a development of unfavorable pH. And, of course, dito na papasok si death phase or the decline phase. Decline or degradation phase. So, there is an increased number of killed bacteria in this phase dahil wala na silang food, dahil ang pH nila is very hindi na stable, so nagkakaroon na ng death phases or bumababa na ang pagkakaroon ng bacterial multiplication. So, this is a bacterial growth phase. The bacterial growth curve ng ating mga microorganisms. Okay, so this is the end of our presentation and I hope you learned a lot. If you have a lot of questions or mayroon kayong mga hindi naintindihan sa ating discussion, please don't forget to write it down to our comment section so that we can answer it correctly. Okay, so please don't forget to like and subscribe to our YouTube channel in order for you to be updated to our next video. So in my next video, I will be teaching you or I will be sharing to you the different methods on how to control your microorganisms. Thank you so much and bye-bye. See you to my next video.